Hello everyone, so today's lecture is reproduction and development. We will focus on human reproductive system. And at the end of this lesson, you should be able to overview the male reproductive organs, especially testes, and explain the structure of spermatozoa. Let's start. So this is the male reproductive system. So what is testis? Testis is a male gonad and it is located and covered in scrotum. So what is the function of scrotum? Scrotum is to maintain the testis temperature, which 2 degrees Celsius below than body temperature. The internal structure of testis consists of many long, highly coiled tubules, called as seminiferous tubule. So what is the function of seminiferous tubule? It is the site where the sperms are produced. In each seminiferous tubule, there are Leydig cells that surrounded by interstitial cells. And the sperm from the seminiferous tubules produced then will pass into the epididymis. So what is the function of epididymis? It is a duct where the sperm complete the maturation and will become motile. During ejaculation, sperms are passed from the epididymis into vas deferens. This is the cross-section of seminiferous tubule. As you can see, the seminiferous tubules are surrounded by Leydig cells. Sperms produced at the center or lumen of seminiferous tubule. Structure of spermatozoa. Spermatozoa is actually the sperms, and it has tiny shape and a tadpole-shaped body about 50 micrometers long. Is it very tiny, right? Spermatozoa consists of a flattened head, a cylindrical body, and an elongated tail. It is produced in the seminiferous tubule, as we learned before, of the testis. Look at the head. The head composed of a haploid nucleus, and the nucleus contains chromatin, consists of 23 chromosomes in human. What about the head? The head also has acrosome. It located at the tip of head, and it contains hyaluronidase enzyme. Hyaluronidase enzyme will help the sperm to penetrate an egg. What about the neck? The neck of a spermatozoa is very short and have one pair of centriole, and it has 9+2 arrangement of microtubules. Meat piece. It contains many mitochondria. Mitochondria provides ATP, so spermatozoa can move faster by beating the tail. What about the tail? The tail function is to move. The sperm will beating its tail to move forward. And the tail contains 9+2 arrangement of microtubules. Thank you. Hello everyone. Today's lecture is reproduction and development. We'll focus on human reproductive system, whereby at the end of this lecture, you should be able to overview the female reproductive organ, especially ovary, and the structure of secondary oocyte. Let's start. This is the illustration of female reproductive system. What is ovary? Ovary is a female gonad. It occurs in a pair, and the function of ovary is to produce the female sex hormones and secondary oocytes. What is secondary oocyte? It is a cell that has size of 100 to 120 micrometer in diameter, and the secondary oocyte consists mainly of plasma membrane and cytoplasm. In the middle, secondary oocyte contain nucleus. The nucleus of secondary oocyte is actually arrested as metaphase 2 during cell division, and it sits inside a cell with a large volume of cytoplasm. Secondary oocyte also contain cortical granules, that small membrane-bounded granules accumulated in the cytoplasm. It just beneath the plasma membrane. Secondary oocyte also surrounded by thick glycoprotein shell called zona pellucida. In this figure, the black line is the zona pellucida. The zona pellucida is a jelly-like coat. Secondary oocyte are surrounded by follicular cells called corona radiata. Corona radiata is made up of granulosa cells of the follicles. What is the function of corona radiata? Corona radiata protect the secondary oocytes as it passes through the ruptured follicular wall. Thank you. Hello everyone. Today's lecture is reproduction and development. Learning outcomes. At the end of this lesson, students should be able to define embryogenesis. Number 2, state the developmental stages from zygote to the formation of morula, blastocyst, and gastrula through cleavage process. Define organogenesis and also state the organ formed from each germ layers during organogenesis. Let's start. What is embryogenesis? Embryogenesis is a process of formation and development of embryo. There are developmental stages from zygote to the formation of morula, morula to blastocyst, and the blastocyst to gastrula. In cleavage, a series of rapid cell division occur during early development. The repeated division increase the number of cells of zygote until it called as blastomere. Solid ball of blastomeres now called as morula. The morula is converted into blastocysts that consist of inner cell mass, blastocele, and the trophoblast. Next, gastrulation. Gastrulation is when the blastocysts become a 3 germs layer. The blastomere cells will divide and rearrange to form ectoderm, mesoderm, and also the endoderm. Organogenesis is a process of formation of organs in body, which involve differentiation of 3 germs layers: the ectoderm, mesoderm, and the endoderm. The 3 germs layer, we start with ectoderm, will give rise to epidermis of skin and also glands, nervous system, tooth enamel, then teeth and pulp, epithelial lining of oral cavity and rectum, cornea and lens of eyes, adrenal medulla, and mammary glands. Next, mesoderm. Mesoderm will give rise to musculoskeletal system, dermis of skin, cardiovascular system, urinary system, lymphatic system, reproductive system, and notochord. While the endoderm will form epithelial tissue lining of digestive tract, vagina, and urethra. The alveoli of lungs form the liver and pancreas, thymus, and thyroid gland. And the last one is neurulation stage. Neurulation stage is referred to the development of dorsal nerve cord. Thank you.